Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Terry King, your host, and today I'm talking to Bethan Littleford Colquhoun, a prize fellow whose research focuses on understanding how animal diets interact with gut microbiomes to shape nutrition, health, and fitness in the wild. Her research sits at the interface of ecology, evolution, and microbiome science. So Bethan, we are really starting to understand the critical role that microbiomes play in health. So for those who don't know, what is the microbiome?
So the microbiome is a really dynamic community of microorganisms which are made up of bacteria, fungi, viruses, archaea, and they inhabit all sorts of environments. They can be found in the air, in the water, in the soil. They can also be found in our homes. But the microbiome that I'm really interested in is the microbiomes that inhabit the gut. So we have microbiomes on our skin, in our mouth, but also those in our gut that help us digest food and play a really critical role in things like immunity and health and disease.
And you're really interested in looking at the microbiomes in animals, aren't you? So what's the overall goal of your research?
So generally, I'm really interested in how environmental change alters what animals eat, so wild animals in particular, but how these dietary shifts change and influence the gut microbiome in ways that then affect things like animal health, sort of survival, but also ecosystem functioning. So I think about environmental change in lots of different ways. So that can be things from urbanization all the way through to things like seasonal shifts, where animals have to deal with changes in food resources as we progress from winter to summer, for instance.
So I know you're using DNA metabarcoding, which is this really powerful method that identifies what animals eat by sequencing the DNA in samples. So talk me through, what is DNA metabarcoding? How does it work? How do you do it?
So DNA metabarcoding is a bit like forensic science. So you can go and collect a sample. For me, a lot of the samples that I analyse are fecal samples. And so these are collected from the environment. They're a really great sample to use because they're non-invasive. So I don't have to interact with animals to go and collect samples. They're in the environment. So I can collect them opportunistically, but I can also do focal studies where I follow certain individuals from a safe distance, watch them poop, and then go in when they've left the area and go and collect those samples from the environment. And so what I do is I bring that sample back to the lab. I can extract all the tiny bits of DNA from that one sample. And from that single sample, I can understand what that animal has been eating, the microbiome of that animal, but also I can understand a bit about the host species as well. So I can think about the genetic fingerprinting essentially and reference those tiny bits of DNA back to a reference database, and that will tell me what species an animal has been eating, what plant species an animal has been eating, what the microbes are in the gut. It's a bit like putting items in a supermarket into your shopping basket and then scanning those barcodes. And if you have that reference library of what's available in the store, you can work out what people are buying. And so it's the same thing, but with fecal samples and plant species and gut microbes.
So it's quite cool though, because rather than kind of having somebody out in the field that's kind of watching an animal, presumably from a distance, because some of the animals you work with are quite big, and you go, right, what are they eating? You're getting quite a big amount of information that's coming out of this fecal sample. It's less of a snapshot and you get more, as you say, it's like a shopping list kind of thing.
Yes, definitely. So what's really brilliant about using molecular methods rather than observational data is anybody can walk into, let's say, a field of cows. You assume that those cows are eating grass, and you wouldn't be wrong probably in that assumption, but it's a lot more nuanced than that. So those cows may be in a field eating grass, but there might be 20 different species of grass in that field. There may be other things that aren't grasses. There may be forbs, so more herbaceous material that those animals are seeking out in the environment. We also don't know if we're just watching a cow, whether they're just taking indiscriminate munches of those grasses or whether they're going in and being a lot more selective. And so DNA metabarcoding really lifts the hood on that. So we're able to work out the breadth of plant taxa that animals might be eating, so those cows, for instance. But we can also start to understand, are all cows eating the same thing? Or are cows separating what they're eating and differentiating what they're eating from the cow that's next to them? So we can really start to dig into exactly what animals are eating and how those resources are being partitioned between individuals and species, which essentially is what determines population dynamics and then ecosystem functioning.
And you've been doing this really interesting series of projects looking at seasonal changes in the diets of animals living in Yellowstone Park in the US. So tell me about that.
Yeah, so that project has been trying to better understand what the large herbivores of Yellowstone National Park actually eat and how they divide up resources across time and space. So that research has been looking at large herbivores from bison, which are these huge herbivores, down to something like a pronghorn, which is relatively small in comparison, and looking at what these animals are eating across different seasons. So Yellowstone is a really interesting environment because in summer you have these really big flushes of green vegetation, whereas in winter a lot of that vegetation dies back and also it then becomes covered in quite deep snow. So we're looking at how that affects what foods these animals are eating, but also the different decisions that those animals are having to make, and looking at the effect that body size has on those decisions. So we found that all species shift their diets from summer to winter, which makes good intuitive sense. But what we actually find that's even more interesting than that is that the individuals of those species make different decisions depending on which season we're looking at. So for instance, in summer, bison form these really big herds of individuals, and those individuals eat pretty similar diets to one another. But in winter, those bison splinter off from one another and their diets then become a lot more individualized. We actually find the opposite for pronghorn. So in summer, diets seem to be more individualized, whereas in winter, they start to form not herds as such, but these bigger groups, and they then become more homogenous in the diets that they have. And this is because of how they interact with the environment. So in winter, bison are able to use their huge heads and push through that snow to access resources that smaller herbivores like pronghorn aren't able to access. And so it's a really neat way of trying to look at how diet influences animal decisions and the nutrition that then is fueling those individuals to get them through the winter.
And your research is doing some quite cool stuff because it's actually overturning ecology kind of classifications that people thought, well, this is exactly what they eat. This is this particular animal. It lives in this particular way. This is another, it lives in this particular way. But actually, your research is showing that that's way too simplistic.
Yes. So we as humans really love to put things into boxes. So coming back to the Yellowstone example, we often refer to bison as grazers. So grazers mean that they're eating predominantly grass. And whilst this isn't incorrect, again, sort of through using molecular methods, we're able to show this is much more nuanced than we ever previously thought. And actually, those classifications of browser versus grazer, for instance, don't apply necessarily to a single species. So some really brilliant research that I was part of at Brown University with a PhD student, Hannah Hoff, she was able to show that rather than classifying species based on that browser-grazer continuum, actually what we can do is start to group animals based on what they're eating across different times of the year. And actually those groups of animals will likely be made up of different species. So applying a single label to a single species really doesn't hold much in the modern conservation, ecology, evolution thinking. It's a lot more complicated than that, and we shouldn't be quick to just assume that a species is either a browser or a grazer, because it could actually be both of those things, maybe in different seasons.
So I know your work is also feeding into conservation efforts with animals that we know are particularly suffering in terms of kind of declining population sizes, so like koalas and giraffes. So what are you looking at and what are you finding?
So conservation is often pinned on legacy techniques, if you would think of it that way. Let's take the koala, for instance, a species that is in huge decline in Australia. It struggles a lot with disease, dog attacks, habitat loss. There's a whole heap of things against a koala, unfortunately. And so many of those populations are in decline. And so the koala is subject to a lot of translocation efforts in order to try and increase the genetic diversity of some populations and repopulate some areas. A lot of those decisions on what animals are moved from location A to location B often depend on the sex of that individual and the genetic makeup of that individual. Research that we've done has shown that although those things are incredibly important, animals must eat to survive. That's not new. The koala is a really special example in that it's a specialist species. It only eats eucalypt. An individual koala can eat anywhere between 1 and 10 different species of eucalypt. But what that means is the koala relies incredibly heavily on its gut microbiome to detoxify that very toxic eucalypt diet, to break it down and extract all the nutrients that it possibly can from that very nutrient-poor food type. And so what we were able to show is that although genetics plays a very important part, and we often think genetics can influence how gut microbiome communities are made up across a species, actually the genetics of a population doesn't match the gut microbiome profiles of individuals from that same population. And if you are to move a koala from location A to location B, although there will probably be eucalypt in that environment, if it's completely different species of eucalypt and a koala isn't primed to digest those new species of eucalypt, it potentially would starve because it's not able to extract all the energy from that very nutrient-poor diet. And so what we were able to show is that we see huge variety and variation of microbiomes across the landscape in koalas. We see these population dynamics as well, so we do see population differences. So it's really not as simple actually as picking up a koala from location A, moving it to location B and saying, it's fine, it has eucalypt, it will survive. It's much more nuanced than that.
So the work you're doing is pretty new, but it's adding a really critical layer of information that can be added to these conservation programs in terms of giving the best possible likely outcomes. And it's based on all of this new, hitherto unknown kind of information that you're now bringing to the picture.
Yes, so this is new work that's being incorporated more and more every day into conservation science. Our limiting factor really has been the methods that we're able to apply in the lab. And so it's only really in the last 15 years that we've been able to even think about generating this data in a very user-friendly, relatively cheap way. So now we're in a really exciting time where we're in an explosion of data. And so we're getting all this data from a single sample, a fecal sample collected in the environment, and we can now add, as you say, these new layers to conservation. We can start really thinking about the wider picture and making better decisions based on better information that's now more readily available for many more individuals within those environments.
So I suppose coming into all of this is looking at the factors that drive this gut bacterial diversity. So talk me through that.
So lots of things are known to influence the gut microbiome. So species identity, so what that host is, whether it's a bison or a pronghorn, the genetic makeup of those hosts, the environment that that animal is found in will also influence gut microbiomes, as well as diet, obviously, and diet is one of the biggest factors that influences a gut microbiome, and a whole host of other things like host health, fitness, but it's not just a one-way relationship. So the host can also influence the gut microbiome, and the gut microbiome can also influence the host. But something that I'm really interested in looking at now is trying to think about whether the gut microbiome should be treated as a host trait. So there's some neat ideas out there looking at whether the gut microbiome and all the genes that it has within the microorganisms that inhabit the gut, whether that offers an adaptive potential for the host. So whether the gut microbiome can adapt and evolve quicker than the host can itself, which therefore then allows the host, the animal, to survive in environments that it previously wouldn't be able to if it wasn't for this gut microbiome. And so I'm starting to think now about whether the microbiome is more an ecological community where bacteria come and go and the functions can change, or whether there is some evolutionary element to it in terms of stability, and whether evolution then acts specifically on the microbiome and the microbes themselves, which then offers this potential adaptive increase within the host itself. So this is a really interesting and sort of growing idea within the field.
And I know one of the other things that you're doing alongside this is you're working on the technological advances in metabarcoding. So what are you working on, and how is that going to help you with your research?
So I'm thinking about two different things predominantly. One is the lab methods that we use. So some work that I did was looking at how we can improve the molecular methods that we use currently for DNA metabarcoding. So at the moment, we're limited by the amount of DNA we can sequence in a single sequencing run, and looking at whether different technologies would allow us to look at a greater amount of DNA within those samples. The greater amount of DNA that we can find within those samples and we can amplify within those samples means that we get more information. So we can start getting down to specific plant species identity, for instance, rather than looking at a slightly higher taxonomic level. We can also start to think about gene transfers and changes in the chloroplast makeup if we're able to target larger sections of the chloroplast genome, for instance. The other thing that I'm looking at is the different ways that we treat that sequence data when it comes back from those sequences. And so what I mean by that is that we have to process that data in order to understand what an animal is eating. But the decisions that we make during that processing will change the end result. So it's like making a cake and changing the quantities of flour that you add will change the cake that you end up with. And it's the same with the decisions that we make when we're analysing this data. So I want to kind of make sure that we're using the best possible processes that we can, and we're making the best possible decisions that we can for every individual system that we're working on. And so I put a lot of time and effort into these less glamorous jobs of just kind of analysing data over and over in different ways and making sure that we're getting the most reliable and actionable research that we possibly can produce, especially because it's being used for things like conservation science and applied ecology, and it goes into these evolutionary theories. And so it's really important that we get it right.
It's basically about data accuracy, isn't
Absolutely.
it? So you're getting loads and loads of different sequences. And I suppose you have to make decisions about, oh, well, we've only got 2 sequences that come from this particular plant. Is that real, or is it contamination, or is it what? So it's that kind of decision making, I'm guessing.
Definitely. And there has historically been this idea in the field that we would remove what we deem rare taxa, so those taxa that maybe only appear a couple of times in a sample. And whilst that might be applicable for one system, it does mean that we're losing information. We don't want to keep data that's false, but we also don't want to throw out data that's true. Because again, it changes the end product, it changes our understanding of ecosystems. And so there's a fine line that we have to toe. And again, it's making sure that we're making the best possible decision. What might be the correct answer for one system might be really different for another. And rather than just plugging into a black box and saying, yeah, I'm just going to run the same pipelines for every sample that comes in, for every project that I work on, it's making sure that we're making those decisions based on real science, real data.
And then that presumably will become kind of a standard for the community as well, to understand, yeah, we do need to keep this in, or no, we don't need to keep that in.
Yes, definitely. And that's what I'm kind of striving for, is not only trying to work out how we best make those decisions, but making those actually more accessible for the wider user. So what I would love to do is to create programs where people can put in the kinds of data that they're using, and we can almost run like sensitivity analyses. So looking at what decisions should you make for your particular data set, so that people can go into their own systems and know that they're making the best possible decisions. And that shouldn't be something that's just held for me. In order for everybody to benefit, and for science to benefit, for conservation to benefit, those tools need to be widely available.
So what's next for you? What areas are you interested in pursuing next?
So I'm really interested in building on the work that I've previously done. So I've been looking, obviously, at diets and gut microbiomes of various species across the world. And so what I'm really interested in doing now is trying to link those things with nutrition. So it's not just about what an animal is eating, it's about the nutritional value that that food offers the host. And so herbivores are really interesting in that their diets are really protein poor, but protein is hugely important for survival. We wouldn't survive without protein. It goes into muscle growth and growth generally. It's really, really important for survival. So for herbivores, if they're not getting that protein from their diet, which is normally how we would get protein, how are they able to survive? How are they producing enough protein in order to reproduce and survive from one year to the next? And there's some really awesome research that has been done in some lab animals, but also in some small mammals, looking at the role that the gut microbiome can actually play in generating this protein, these essential amino acids that a host is not getting from their food. And I would really like to start applying that to the systems that I work in.
Bethan, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our social media channels. For more information about the Milner Centre for Evolution, you can visit our website.